Hello, and welcome to EMP News with Rosy, Jodie, and Michaela. Today, we have a few subjects to talk about, but first, we need to lay down some vocab for you. This may get a little complex, but stick with us on this. Firstly, we'll go for atoms, protons, neutrons, and electrons. An atom is the smallest part of an element. These are made up of protons, neutrons, and electrons. The centre of the atom is made up of protons and neutrons, more commonly referred to as the nucleus. The electrons can then be found orbiting this nucleus. We will also come across biomolecules. All of the following are carbon-based that will be touched upon throughout this broadcast. We have carbohydrates, used to store and supply energy, proteins, used for transport and structure, and finally lipids. These are fats within cell membrane and are used for insulation and protection, signalling molecules, and for minor energy supply. Finally, we have monomers and polymers. Monomers are the building blocks of a polymer. Think of this like a Lego car, for example. The car is the polymer, the final product. The Lego bricks are monomers, the things that build and join together to create the final product. Now, on to biomolecules. We have three main biomolecules that are commonly used within biology. We'll start with carbohydrates. As we can see from the images, carbohydrates are fairly simple polymers made up of monosaccharide monomers. These are made up of carbon, hydrogen, and oxygen in a hexagonal bond. These are more commonly known as glucose, fructose, and galactose. There's a word you may recognise, glucose. This is the most widely known monomer that is used to create polysaccharides. Polysaccharides are a substance that are created when these glucose molecules from glycosidic bonds and form together to create a chain of monomers. These chains are used for storage and can be broken down by using hydrolysis. A very well-known polysaccharide is called glycogen. This is found in the animal body, specifically the liver and muscles. Here, they get broken down into glucose. Finally, we'll briefly go over proteins, as we have a whole section dedicated to this topic later in the broadcast. Proteins are still similar in the elements that they're made up of, carbon, hydrogen, and oxygen. However, proteins also include nitrogen. Proteins are mostly used within muscle building. Think of bodybuilders and all the protein shakes they drink. But proteins also aid within the immune system. They also work within enzymes. Proteins are actually very clever and are widely used within the body. They will change shape dependent upon the function that they are due to serve. Now, onto lipids. Lipids are more commonly known as fats. A little example of these are butter, oil, cholesterol, and steroids. These are made up of carbon, hydrogen, and oxygen, just like carbs. However, the structure is slightly different. There are two bases to lipids, a backbone of glycerol, with fatty acids bonded to this. When we think of fats, most people think these are a bad thing, but our bodies need certain fats to survive. This is because they store energy for a long time. Take seals, for example. When they're young, they're all fluffy and cute. This is because they need the fur to stay warm. However, when they're not so fluffy and cute as they get older, this is because they have a lot of fat stored, so they can conserve their energy in the extreme weather conditions they live in. This is called blubber, and this fat is essential for their survival. As mentioned at the start of the broadcast, a polymer is essentially multiple monomers that have bonded together. Think of this like building a house. Monomers are the bricks, and a polymer is the finished house. For example, monosaccharides create carbohydrates, lipids are made up of fatty acids, and proteins are made up of amino acids. The process of joining these different monomers together is called polymerization. 2 monomers joined together is called a dimer, 3 joined together are called a trimer, and 4 or more bonded monomers are called a polymer. To break down polymers, we use the process of hydrolysis. This is where water is added to the polymer, causing the bonds to break down and separate. This recreates the makeup of the whole substance. Within biology, hydrolysis is mostly commonly used within ATP to enable the metabolism, but we'll delve further into that later.
Let's take a look at enzymes. What are they? What are their structures? And how does this structure affect the function? Starting from the beginning, what is an enzyme? An enzyme is a biological catalyst. It speeds up already existing chemical reactions. The great thing about enzymes, they don't get used up in the chemical reactions, therefore they can be used again and again. Enzymes are a funny sort of shape, often referred to as similar to Pac-Man. As you can see, you have the head of Pac-Man and the mouth. The mouth, or better known as the active site, is where a substrate comes to bind together with the enzyme to create an enzyme-substrate complex. But we'll talk about that more in depth later on. Let us look inside the enzyme. What actually makes it up? Well, enzymes are almost always proteins. There are 4 structures to a protein in order to make it functional. Firstly, we have the primary structure. Starting off really small, we have the amino acids. These are held together with peptide bonds, creating a long string of amino acids called a polypeptide chain. DNA determines the particular sequence and structure of these polypeptide chains. The sequence of the amino acids is crucial for the structure of the protein. If we go even smaller into the amino acids, we'll see that they each have a carboxyl group on one side, an amino group on the other, and at the bottom a variable R group. Next, we have the secondary structure. This is when the polypeptide chains begin to fold. The 2 most common folding patterns that we can see are the alpha helix and the beta pleated sheet. The type of the folding that occurs is dependent on the sequence of amino acids. Hydrogen bonds are key to this folding process. In the tertiary structure, rather than the hydrogen bonds, this time the variable R group is the cause for the next level of folding. In each amino acid, the R group can vary. It can act as the remote control of the amino acid, making it act a certain way. The R groups can have different properties. For example, some may be hydrophilic, meaning they are water-loving, and some may be hydrophobic, which means that they don't really like water. And so, as the proteins begin to fold, the hydrophilic R group will be on the outside of the structure, surrounded by water, and the hydrophobic types will nestle themselves inside the structure to stay away from the water on the outside. Other factors can cause the folding to occur: ionic bonds, hydrogen bonds, and disulfide bonds. Some inhibitors can be feedback inhibitors, and a feedback inhibitor that I have come across before is FIL. No, not Phil as in Philip. FIL as in feedback inhibitor of lactation. FIL is a polypeptide that accumulates in breast milk produced by the Acini cells. It stops milk production when breasts become too full, giving the impression that too much milk is being produced. Just note that prolactin and lactocyte are 2 very important factors in milk production. Prolactin is the hormone responsible for lactation, and lactocyte is a milk-producing cell. When the breast is full of milk, the walls of the alveolus expand and stretch, altering the shape of the prolactin receptors, so that the prolactin cannot enter via those receptor sites. Similarly to non-competitive inhibitors that sit at the allosteric site of an enzyme and cause the enzyme to change shape, therefore making the active site unable to be used, or for a substrate unable to bind. Milk synthesis cannot occur without the presence of prolactin, the hormone, and on the walls of the lactocytes are prolactin receptors. These receptors allow prolactin in the bloodstream to move into the lactocytes and stimulate milk production. As the breast starts to empty of milk, the prolactin receptors return to their former shape and again allow prolactin to pass through, therefore increasing the rate of milk synthesis.
Enzymes will speed up already existing reactions. They do this by reducing the amount of energy actually needed to kickstart the reaction. This is known as activation energy. Think of activation energy as a barrier or a mountain. Before the reaction can start, the mountain needs to be climbed or the barrier overcome. But it needs to be done by using as little energy as possible. Imagine that the substrate is one side of the mountain and the product at the other. Energy in this case is heat, and enzymes allow reactions to take place at lower temperatures such as the human core body temperature.
Enzymes do this by being highly specific shapes that allows the substrate to fit directly into the active site, therefore speeding up the reaction. Ever heard of the lock and key model? Thanks to Dr. Hermann Fischer, who, in 1899, the lock and key model was first theorized. This suggested that the enzyme and the substrate are specifically complementary geometric shapes that fit exactly into one another, meaning only the specific shaped key, in this instance the substrate, fits into the hole, this being the active site, as shown in this picture. Do you remember when you were a child and you played with wooden shaped puzzles and how only the exact shape would fit into that space it was intended for? It didn't matter how many times it was rotated until the shape perfectly matched, it would not fit. Well, that's the same with the lock and key model. Only very specific substrates fit into the enzyme perfectly.
In 1958, Dr. Daniel Koshland created a modified version of the lock and key model, calling it the induced fit model, where he discovered the interaction between an enzyme and substrate doesn't necessarily need to be a perfect match. The active site of the enzyme conforms until the substrate is completely bound to the enzyme. This then activates the enzyme into performing its function. In basic terms, the active site isn't a perfect match for the substrate, therefore ever so slightly adapts its shape to fit in with the substrate. For example, when you put on a pair of jeans that's wet and slightly tight, they don't quite fit, so you need to wriggle and force them to fit and do the job intended, just like the active site. Even though they are very similar regarding their findings, they are very different in many ways. The lock and key model says it needs to be perfect for the enzyme to work in its best potential. However, the induced fit model argues that it doesn't need to be perfect and it can adapt for the enzyme to work. After all that information, you'll be a genius in no time. Thanks for listening, folks, and EMP News will be back for another broadcast soon.
In 1958, Dr. Daniel Koshland created a modified version of the lock and key model, calling it the induced fit model, where he discovered the interaction between an enzyme and substrate doesn't necessarily need to be a perfect match. The active site of the enzyme then conforms until the substrate is completely bound to the enzyme. This activates the enzyme into performing its function. In basic terms, the active site isn't a perfect match for the substrate, therefore ever so slightly adapts its shape to fit with the substrate. For example, when you put on a pair of jeans that are wet and slightly tight, they don't quite fit. You can wriggle and force them to fit and do the job intended, just like the active site. Even though they are very similar regarding their intentions, they are very different in many ways. The lock and key model says it needs to be a perfect fit for the enzyme to work to its best potential. However, the induced fit model argues that it doesn't need to be perfect for the enzyme to work.
Three external factors which can affect enzyme activity include the temperature, pH level, and substrate concentration. The activity of the enzyme gradually lowers as the temperature rises more than the optimum temperature, until it reaches a certain temperature at which the enzyme activity stops due to the denatured protein. pH levels specifies the acidity or basicity of an aqueous substance. Each enzyme has its optimum pH at which it works best. Small changes can slow down the rate of a reaction, whereas larger changes can cause the enzymes to be denatured and stop working. Substrate concentration is the rate of a chemical reaction increases with the concentration of substrates. The more the substrate molecule, the more active sites in contact with the substrate, which means an increased reaction, eventually increasing the substrate concentration, will have no effect as the enzymes are already working to their maximum capacity.
And after all that information, you'll be a genius in no time. Thanks for listening, folks, and EMP News will be back with another broadcast soon.